Hi everyone, today we're going to talk about alignment and phylogeny. This is a topic that might already be familiar to you, and if that's the case, then hopefully this is a good refresher, or maybe I will go into a little bit more depth than you're used to. And you're also free to just fast forward and see if there's anything new. But for I think a lot of you, I'm sure have heard the words alignment and phylogeny and phylogenetic trees, but they might seem very mysterious to you. And so my goal is to show you just enough of the mechanics and details of how you make a phylogenetic tree so that it doesn't seem as mysterious to you anymore. The reason why I think this is an important topic for this course is that it's an important concept for understanding how environmental DNA sequences of microbes are classified into taxonomy. And as we have already seen with the first two papers we've discussed in this course, making colorful bar charts of microbial taxonomy is like the first figure of every single microbial diversity paper, right? And there are two different strategies for generating these charts of bacterial taxonomy. One is you compare your environmental sequences to some sort of database. And this is exactly what we did with the BLAST activity that we did last week. You took your mystery sequence, you compared it to the NCBI non-redundant database, you got a result, and you could infer a taxonomy of your sequence, or you could not infer a taxonomy, which is also a result. In practice, you wouldn't normally use the NCBI non-redundant database because it has all of the sequences in the universe that have been sequenced. Whereas if you are really doing this experiment, you already know that you have 16S ribosomal RNA sequences, so you would use a database that is specialized for that purpose, and there are multiple options of such databases. Okay, so that's how you do this. You just compare it to database, you get a result, and you make some sort of decision like, if the match is 85% similar, then I will count that as being good enough, and that will show up in my result. It's a little bit more complicated than that. You can make different decisions for the species level versus the genus level versus the family level, etc. But you basically have to come up with some kind of arbitrary threshold by which you decide this is a hit and this is not a hit. And that's how you make a chart like that. The other strategy is to use phylogeny. And this is the gold standard in biology. If you've ever taken an intro biology course, someone at some point said evolution is the only way that biology makes sense, and phylogenetic trees are the way that we display evolutionary information. So evolutionary history is the gold standard to determine taxonomy. The problem is that it's really hard to use phylogeny to determine taxonomy, especially when you're using sequences that are very short, which is usually the case. Because the DNA sequencing technologies that we use today generate very short sequences. And short sequences are not good for phylogeny. Even if you do have good long sequences, it's still difficult to interpret a very large tree, which is what you have to make if you're going to make a figure that is describing all of microbial diversity. That's a very large tree. So if you want to make an overview chart of all of the diversity in your sample or your system or whatever you are studying, a tree is just a really hard way to do it. So this is an active area of research. There are some clever strategies out there that take the best of both worlds, that combine these two strategies with database comparisons and phylogenetic trees in various ways. But that's cutting edge experimental stuff. And for the most part, you got to choose one of these two. And right now, the database way is the easy one, and therefore it's the most popular one. But the phylogeny is clearly better, and that's why we're going to talk about it today. So first, a really fast intro to phylogeny. This and the next few slides are slides that we use in Biology 1620. So for those of you who have already taken that course, this should be a refresher for you. Although maybe the slides were changed slightly in your version of the course, but we definitely went over monophyletic, paraphyletic, and polyphyletic. And if you don't remember the differences between those three words, then you're not alone. I also have to remind myself, especially what is the difference between paraphyletic and polyphyletic. It's not so important. What's really important is that monophyletic is the correct one. Okay, what does this mean? Monophyletic means that all of the members of your group, a monophyletic group, contains species that are all descended from a single common ancestor. Paraphyletic and polyphyletic groups do not match this criterion. They do not follow this rule because, for example, in paraphyletic, a group that includes L, M, and N but not O is paraphyletic because it does not include all of the descendants of this ancestor C. It's not including O. And polyphyletic is does not follow that rule because if you have a group that includes H, I, J, K, L, and M, then L and M do not share a unique ancestor with these species because it is descended from C, and you're not including N and O. And so this is a polyphyletic group. You have multiple ancestors of the species that are in your group. So these are not correct groupings for making taxonomic classifications. Taxonomic classifications should reflect monophyletic groups. All of the species should be descended from the same ancestor. So this is actually a pretty tricky concept. It is simple in principle, which is why it's a good principle. But when you start looking at lots of examples, it can have surprising and confusing consequences. And the most famous example of this is that of birds. So for a long time, we grouped birds separately from the reptiles, like crocodiles and lizards and turtles. With more information, we, especially with DNA sequencing, but even before DNA sequencing, there were hints that the correct phylogenetic tree looked something like this, where crocodiles and birds are sister groups. That is, they are very similar to each other and share a recent common ancestor. And so there is no way to draw a taxonomic classification with this phylogenetic tree such that birds form their own unique group separate from any of the reptiles. You can say that birds are birds, which is tautological, that is, it's just circular reasoning. Yes, birds are birds, but they share a recent ancestor with crocodiles, a more recent ancestor with crocodiles than the crocodiles do with any other reptiles. So actually the important point is that crocodiles are more similar to birds. That is, they share a more recent common ancestor with birds than they do to lizards, snakes, and turtles. So if you want to say that crocodiles are a reptile, then you also have to say that birds are a reptile. Or you can say that crocodiles and birds are something else and the reptiles are here. But then actually, according to this particular tree, you'd then have a problem because lizards and snakes would have to be their own group and turtles would have to be their own group, and then reptiles are just fictional. There is no such thing as reptiles in that case. So the point is, you can make up whatever words you want. In some ways it doesn't really matter. Like biology doesn't care what words you use. But what is important is that when you do make groups, the groups should be monophyletic. In this case, if you want to call something reptiles, then you have to include birds in that group called reptiles. Okay, I think have succeeded in taking a concept that you thought was simple and making it more complicated. So let's go over some more examples. These are, again, examples from 1620, but they're good examples, so we're going to use them again. I'm not going to actually make you answer these questions. I'm just going to go over them quickly. So, for example, frogs, salamanders, and caecilians. Is that a monophyletic group? Yes, they all share a common ancestor. You are not including anything that you should not, and you are not excluding anything that you should not. Do lizards, snakes, and turtles form a monophyletic group? No, because they share a common ancestor here, and there are additional descendants of that ancestor, crocodiles, alligators, and birds, that you are not including in this group. So that is not a valid taxonomic group. You cannot make a taxonomic group of lizards, snakes, and turtles that does not include crocodiles, alligators, and birds. Do turtles, crocodiles, alligators, and birds form a monophyletic group? Yes, because they all share a common ancestor, and you're including all of the descendants of that ancestor. Do ray finfish, coelacanths, and lungfish form a monophyletic group? No, because their common ancestor is here. And in order to include those three groups in a larger monophyletic group, you would have to include all of the descendants of that ancestor, would be all of the amphibians and sauropsids and mammals as well. all of these in a monophyletic group? No, for the same reason. Their most recent common ancestor would be all the way back here. And so to put all of these in a group, you would have to include everything on this tree. Right? So there are rules for what you can say is a valid taxonomic group. It's okay if you didn't quite catch all of the details of those. Try to think through it, but again, I'm not going to quiz you. The important principle is that a true taxonomic group is a monophyletic group. Okay, now moving on to how do you make a tree in the first place? In those cases, we were assuming the tree is correct, but how do you know if a tree is correct? So in order to evaluate trees, we need one more concept, which is that of ancestral versus derived traits. So here is another phylogenetic tree, here describing different adaptations during the evolution of fish and mammals and lizards, where during evolutionary time, these different adaptations arose in these different groups, and evolutionary biologists could think through this and using fossils also make a tree that shows when these adaptations arose. The ancestral traits are those that were present in the ancestor, and the derived traits are the new ones that arose in the descendants. So derived traits come after ancestral traits. Ancestral traits are the original state, derived traits are the new ones. Okay, so just a bit of terminology. You could do the same thing with DNA sequences. So this is getting to the point of this course. You can make a phylogenetic tree instead of with morphological traits, like whether you have a backbone or not. Instead, you can make it according to whether you have an A, C, T, or G in each position, such that you have, instead of traits, you have sequences. And so the ancestor has a particular ancestral sequence, and then the descendants have derived sequences like this. So here's one example, and you can draw out this tree of exactly when each base at each position changed during the evolutionary history of these species, as represented by their DNA sequences. And so at each position of the DNA sequence can be considered like one trait. So instead of does it have a backbone or not, it is does it have an A, C, T, or G in that position. Instead of does it have hair, it can be does it have an A, C, T, or G in that position. Here's an example that we use in 1620, and here is another example that I like to walk through to explain how it is that you construct a tree in the first place. So here, again, someone wrote out a tree for us, and we can see how the tree explains the present species and how they evolved from an ancestor. But how do you draw a tree in the first place? That's what we want to know. Okay, so here's some examples. We sequenced DNA from 5 different species. The DNA sequence is extremely short because I needed to have a short sequence in order to explain this as an example. So the way you do this, and this is literally how a computer program would do it if you were using one to make a real tree from real data, is that it looks at one position at a time. So this is called an alignment, first of all. An alignment is all of the DNA sequences lined up so that you can compare one position at a time. So if we look at the first position, we see that humans, Romulans, and Vulcans have an A in this position. Klingons and Ferengis have a C in that position. And so one way you could explain the observed data with a tree is if A was the ancestral state, then it changed to a C in the part of the tree leading to the Klingons, and it also changed to a C in the part of the tree leading to Ferengi. That's just one tree. You could try a different tree. What if you rearrange the species on the tree like this, such that C was the ancestral state? It changed to an A in this part of the tree, and then the A changed back to a C in this part of the tree, and it changed to an A in this part of the tree. That is another tree. How do you know which one's better? How do you know whether this one is better or this one is better? One principle that you can use to decide between alternative trees is called parsimony, which is that the simplest explanation, all things being equal, is the best one. This is also known as Occam's razor. It's not the only way to decide which tree is better, but it's the first one that we always teach about, because the other ones are more complicated and more based on statistics. Okay, so you would say that this one is worse because it involves more changes. You have to, the C has to change to an A and it has to change back to a C, and that does not seem as simple as just one change from A to C. Okay? So you keep doing this for each position. We can do this for the second position. We could draw the tree such that it starts with a C and you have two changes, a change to a T here and a change to a T there. Third position, same thing. We could draw the tree so that you have a G changing to a T here and to a T here, and so on. You do this for each of the positions, and for each tree that you draw, you can map out all of the changes onto the tree. So for the examples that I just stepped through quickly in these slides, in total for this tree, for all positions, we had 11 changes, a C to a T in this part of the tree, these 4 changes in this part of the tree, and so on. 11 changes, and it also predicts what is the sequence of the ancestor. And this is one of the super powerful things about phylogeny in general, is that it not only provides a possible explanation of how the species alive today evolved from an ancestor, it also makes predictions about that ancestor. And when you're dealing with DNA sequences, it predicts the actual sequence of the DNA of that ancestor. And there are some experiments where people have actually done this, and they have where they've actually used that prediction to synthetically make the ancestral DNA sequence or the ancestral protein sequence, and then study it in the lab and ask if the properties of that protein, that predicted ancestral protein, for example, have properties that you would expect based on the evolutionary history. So I can point you to those papers if you're interested in that sort of thing. It's pretty cool because it's a great merger of evolutionary theory and molecular biology. But anyway, back to this example here, we have 11 changes, and I could assign you a homework. Can you draw this tree by shuffling the relationships among the species? Draw a different tree that is better than this one, that is, has fewer changes than 11. And I did that for you, and I came up with one that has 8. And maybe if you really want to, you could try to see if you can come up with one that's better than 8. So if this one is the best that our computer program could find, then we might settle on this tree as the one that we publish in our paper, including the statistics of how much better this tree was than all of the possible alternative trees. So that's my super simplistic explanation of how you make a tree. Again, the logic that I was using is called parsimony, which is only one way that you can use to evaluate how good a tree is. In practice, we do this for obviously a lot longer sequences and more sequences. We often make trees with thousands of sequences. And what you do is you just make like millions of different trees and then use a computer to evaluate all of the possible trees and use some fancy statistics to decide which one is the best. Or to decide, do any of the trees make sense, or maybe there is no good evolutionary history for this particular collection of sequences, right? Sometimes the answer is that, no, I can't make a good tree with this data, something's wrong, right? Okay, that is my super fast summary of phylogeny. Again, this is important because this is one important way to assign taxonomy to microbes. All right, email me if you have questions, or we can discuss it in class, and I will see you next time. Bye-bye.